So first, let's discuss the normal synthesis, function, and regulation of parathyroid hormone, which is the main hormone produced by the parathyroid gland. So most individuals have 4 parathyroid glands, which are shown in this picture as 4 tiny yellow glands behind this pink thyroid gland. And usually there are 2 parathyroid glands behind each lobe of the thyroid. Each parathyroid is only about 3 to 4 millimeters in diameter and weighs about 30 grams, which is somewhat comparable to the size of a grain of rice. A cross-section of one of the lower parathyroids in this first image is enlarged in the middle to show the histology. And the white round cells are fat cells, which make up between 25 and 40% of the cells within normal parathyroid tissue. And the small dark regions are further magnified in this image, and these cells are the chief cells, which are the cells within the parathyroid that secrete parathyroid hormone. This simplified image shows the main steps in PTH synthesis in a chief cell. And so the initial precursor is a 115 amino acid polypeptide called pre-pro-PTH. Pre-pro-PTH is cleaved at the N-terminal portion first to pro-PTH and then to PTH, which consists of 84 amino acids. PTH is also known as intact PTH, and this is the form that's released then into the circulation, represented here by the red blood vessel. Intact PTH is also the bioactive form of PTH, and it's the form that's detected by most current PTH assays. In addition to intact PTH, some inactive carboxy terminal fragments are present in the parathyroid gland, and sometimes these are released into the circulation as well. So the parathyroid can vary the relative amount of active and inactive PTH that's secreted based on the body's calcium status. Now let's look at an overview of the function of secreted PTH. Once in the bloodstream, PTH has 3 main target organs or sites of action. The 3 sites are the bone, the kidney, and the intestine. So first, let's look at how PTH is involved with these organs in regulating serum calcium. So PTH binds directly to the cell surface PTH receptors on the bone and the kidney. And the PTH receptor is a member of the G protein coupled family of receptors. In the bone, PTH binding causes an increase in bone resorption by activating both osteoblasts and osteoclasts, thereby increasing calcium release from the bone into the bloodstream. In the kidney, PTH binding causes increased calcium reabsorption in the ascending limb of the loop of Henle, as well as in the distal tubule and collecting duct, thereby directly increasing the calcium levels in the bloodstream as well. PTH does have a second action in the kidney, which is to increase the formation of active vitamin D within the kidney. Once formed, this active vitamin D travels to the intestine, where it promotes increased absorption of dietary calcium. So this is why PTH's action on the intestine is considered indirect, because PTH doesn't actually bind directly to cells here, but instead it promotes the formation of vitamin D, which does act at the intestine. So the main effect of PTH via its 3 main target organs is then to increase serum calcium. In addition to regulating serum calcium, PTH also functions to regulate serum phosphorus. And let's look again at those same 3 sites of PTH action, the bone, the kidney, and the intestine, to understand how PTH affects serum phosphorus. So at the bone, the same effect of PTH to increase bone resorption will release phosphorus into the bloodstream just as it did with calcium. And this is because the main minerals in the bone are calcium and phosphorus. Then at the kidney, PTH binding to its cellular receptors in the nephron will increase the excretion of phosphorus into the urine. And remember that this is the opposite of PTH's effect on urinary calcium. The second function of PTH on the kidney is identical to what we just saw, which is to promote the formation of active vitamin D. And this activated vitamin D, just as we saw before, will act at the intestine to increase dietary phosphorus absorption as well as that dietary calcium absorption. So interestingly, even though PTH acts to increase phosphorus in the bloodstream via its direct action at the bone and its indirect action at the intestine, the predominant effect of PTH is to decrease serum phosphorus. And why is this? Well, the hypophosphatemic or phosphate excreting effect at the kidney usually prevails over the other effects at the other organs. And the serum phosphorus regulatory effect of PTH is considered a secondary effect since its main or primary effect is to regulate serum calcium. Now that we've discussed PTH synthesis and function, understanding the regulation of PTH should be relatively straightforward. And so calcium, as you might expect, is the main regulator of PTH via a negative feedback loop as shown here. So if there's insufficient calcium in the bloodstream, the negative feedback on the parathyroid gland will be reduced so that PTH can be secreted in increased quantities. Here's a more detailed depiction of exactly how serum calcium regulates PTH secretion. So calcium, which is represented in this picture by the green triangles, binds to a calcium sensing receptor on chief cells of the parathyroid gland to regulate PTH. And on the left side, there's abundant calcium. So that calcium sensing receptor can sense a lot of substrate and reduce the amount of PTH secretion. But on the right side, there's relatively little calcium. So when the calcium sensing receptor doesn't sense any substrate, the parathyroid secreting cell, or the chief cell, is stimulated to make more PTH. PTH is also regulated by phosphorus, but phosphorus is a lesser regulator of PTH, meaning its effects are generally outweighed by the effects of serum calcium. Phosphorus regulates PTH via a positive feedback loop rather than a negative one, and that means that increasing the levels of phosphorus in the bloodstream will stimulate PTH to be secreted, which in turn will act to decrease the serum phosphorus back into the normal range.